Our next speaker is Alon Kahana.
Good evening. It's not advancing. Not working. Well, my lab studies also a regeneration model using adult zebrafish. And I have a small lab. Most of the work today was done by a very talented postdoctoral fellow, Alfonso Serra-Villa, who is finishing his postdoc with me and will be looking for a job in the coming year. Just wanted to let you know. And also, with his departure, I will be looking for a couple of postdocs to replace him. So if you're interested in the work and you're a graduate student, seek me out. So what I'll tell you in the next 12 minutes is that adult zebrafish extraocular muscles can regenerate, and the regeneration is driven by de-differentiation, a reprogramming of the cell at the cellular and nuclear genetic level. Autophagy is induced to clear the sarcomeres so that the myocyte that's multinucleated, syncytial, and filled with the sarcomeric machinery can become a proliferating myoblast. FGF signaling is important for the activation of autophagy, as well as inducing other signaling cascades that include caspase-3 functioning in a non-apoptotic pathway. Blocking autophagy or FGF signaling blocks regeneration. And finally, if I have time, I'll talk about our transcriptome analysis, which reveals remarkable similarities between the reprogramming and cancer stem cells in humans. Degenerative diseases lead to cell loss. That's basically their function, their hallmark. And in order to repair these degenerated tissues, whether it's by trauma or disease, we need to replace cells. Now, differentiation, making the myocyte in the textbook, is always one-directional. But we know that we can actually reprogram cells to become more embryonic-like. The problem with using this therapeutically is that the cells that are now induced to become pluripotent still need to re-navigate that entire process before they can become differentiated cells again. And therapeutically, that's a very difficult process. I believe that the real solution to regeneration of tissue that's been damaged through disease or trauma is localized reprogramming, where cells maintain their lineage restriction and can proliferate, reform the muscle mass, and then redifferentiate. So how do we figure this out? This is the model that we're using. There are 6 extraocular muscles around each eye. In the adult zebrafish, we do a myectomy, meaning excise approximately 50% of the muscle. And then in 7 to 10 days, the muscle reforms. And we have actin GFP so that you can see the muscle in real time. This was all published approximately a year ago in Investigational Ophthalmology and Visual Sciences. Just to briefly characterize what we do to show regeneration, this is a craniectomy in an adult fish where we lift the brain, expose the extraocular muscles, and you can see the muscle right there. This is the right lateral rectus muscle. We cut this part, the distal half, leaving a proximal half, and then over the course of several days, the distal half reforms. And this is a growth curve that we can do to show this. It turns out that following myectomy, the residual stump becomes highly, highly proliferative. The robust proliferation that occurs throughout the residual stump is quite remarkable. And when we looked histologically at what happens in the proximal stump, we found that the cells become very mesenchymal-like. And our publication showed activation of mesenchymal markers, showing that these myoblasts are amorphous, begin to proliferate and migrate to reform the lost muscle tissue. So the question is, where do these cells come from? And again, to very briefly restate what's been published, this is not driven by PAX7 positive satellite cells, which we can see in the embryo, and we can see in the adult somatic muscles, even though very rarely. But in the adult extraocular muscles, we do not see PAX7 positive satellite cells. The qPCR is at the limits of detection, and with electron microscopy, we don't identify classic satellite cells in adult extraocular muscles. We also did multiple types of experiments to do lineage tracing, and this is one of them, where we cut the muscle, labeled it with BRDU, waited 30 days, cut the muscle again, labeled it with EDU, and looked to see whether we had double labeled nuclei. And you can see here the elongated nuclei that are labeled with both BRDU and EDU. And this is just a snippet of what we've published, showing that these nuclei can dedifferentiate, regenerate, redifferentiate, and then again dedifferentiate, proliferate, regenerate, and redifferentiate. And finally, with Dextran fate mapping, after we cut the muscle, disrupt the plasma membrane, Dextran fluorescein can enter, get trapped, and here is our experiment showing that these trapped Dextran fluorescein in the myocytes eventually regenerate into the complete muscle. So this is just a brief summary of what's been published. And the question is, how does this actually happen? What's the mechanism? And first of all, the question is, you have this multinucleated syncytial myocyte that's filled with a sarcomeric apparatus, and we hypothesized that autophagy, which is important in muscle disease, in this context is important in disassembling the sarcomeres. Using LysoTracker, we show that there is induction of acidification in the autophagosomes. Oh. Whoa. We also, using chloroquine for inhibition of autophagy, as well as using chloroquine to block autophagy, we show that there is a delay in regeneration. Oh. Let me see if I can find where we're at. If I start running out of time, I'll just skip some things. Using LC3, which changes from LC3-1 to 2 as part of the autophagy flux, we show that autophagy is induced. We have zebrafish that have GFP-LC3, where there's punctate staining when autophagy is induced, and we see that as well. And finally, by electron microscopy, not only do we see the autophagy, but we actually see the sarcomeric remnants within the double membraned organelles of the autophagosomes. What's interesting is that when we block autophagy, we end up getting accumulation of nuclear debris that we initially thought was apoptosis. But it turns out that it's not apoptosis. As a matter of fact, caspase-3, which is induced during regeneration after injury, I representing injury, if we block autophagy, caspase-3 actually goes down. And what we are actually seeing is non-nuclear. So you're looking at tunnel positivity that does not correlate with DAPI staining. And it was electron microscopy that clued us in to the fact that these are actually the excess nuclei. And this is how the regenerating myocytes are dealing with their excess nuclei. How is autophagy activated? We use laser microdissection to look at the transcriptome, and we specifically tested genes that are involved in the autophagy pathway. And Beclin-1 and ATG5 are two genes that seem to be induced in the process. And so then we used both chloroquine and morpholinos against ATG5 or Beclin and tested whether autophagy is actually important in extraocular muscle regeneration. And the answer is yes. So this is a normal muscle after all of this part was removed. You can see that there is elongation and regeneration of muscle. Whereas if you block with chloroquine, the muscle does not regenerate. Likewise, if the gray line represents this half point. If we block autophagy induction by blocking Becklin or ATG5 expression, we likewise get blockage of regeneration. Now, what are the cellular signals that might be involved in inducing autophagy? So we know from the regeneration as well as embryogenesis literature that FGF signaling is very important. We tested to see whether FGF signaling is important for extraocular muscle regeneration, and we were not surprised when we found that indeed it is. Whether we're using drugs against FGF signaling or a dominant negative FGF receptor that's induced by heat shock, we see a dramatic delay in the regeneration of FGF, of the regeneration of extraocular muscles when FGF signaling is blocked. Furthermore, it turns out that proliferation is significantly reduced, suggesting that if you consider the ability of a myocyte to dedifferentiate to a myoblast and proliferate to be the hallmark of dedifferentiation, FGF signaling is important for that dedifferentiation process. We further looked at the MEK-ERK signaling pathway and showed that this is the pathway that seems to be involved in FGF signaling to the cell. Now, furthermore, we looked at different aspects of regeneration that we found to be important, and today I'm telling you about autophagy, so I'll stick with that. And if we block FGF signaling, it turns out that we block LysoTracker staining and we block autophagy. So here is accumulation of P62 sequestosome, looking at the flux of autophagy along with a blockage in the LC3-2 accumulation. We then looked at the pathway, specifically looking at the different phosphotypes of ERK1 and 2, and it turns out that it's ERK2 that seems to be important. And here we're looking at ERK1 and total ERK as well as ERK2, phospho-ERK2 and total, and ERK2 seems to be induced very early, within 24 hours after the injury, suggesting that ERK2 is the primary signaling pathway of FGF signaling. Going back to the caspase-3, which I mentioned earlier, we showed that caspase-3 activation by the injured extraocular muscles is not important in apoptosis, and instead we believe it's actually important in degrading other aspects of the cell, maybe cell cycle related genes, maybe inhibitors or regulators of gene expression. And we're investigating this now. And these papers, this paper on FGF was just published. The paper on autophagy is in press in the journal Autophagy. And then in the last few seconds, I'll just mention what we're doing in the nucleus. So obviously you have a nuclear reprogramming in addition to the cellular reprogramming. So we did a complete transcriptome analysis with ribosomal RNA depletion and looked at the transcriptome. And what we found and focused on are a series of genes that seem to be important for my clinical practice in cancer. And many of these genes actually have drugs that are in trial for treating different types of cancer. And so in collaboration with a colleague who studies breast cancer stem cells, we looked at the concordance between the transcriptome of breast cancer stem cells, either CD44 positive, CD24 negative, or ALDH positive cancer stem cells. And the concordance is actually quite remarkable between zebrafish myocyte dedifferentiation genes and human breast cancer stem cells. And breast cancer is a particularly concordant one. Colon cancer is not, which is very interesting. And so in summary, what I've hopefully shown you is that zebrafish can regenerate their extraocular muscles. And this is a unique model in that the entire proximal stump dedifferentiates in a fairly uniform fashion. So we have a population of fairly homogeneous cells that we can study at the genetic, epigenetic, and cellular level using modern molecular tools. And then finally, there's a connection between embryogenesis, regeneration, and cancer that we are beginning to probe. And thank you very, very much for your attention. Sorry about the technical difficulties. Oh.
I just, when you were talking about the concordance between the embryonic expression versus the breast cancer stem cells, and you mentioned that you also sorted based on ALDH positive, so the aldofluor assay, I'm presuming.
So aldehyde dehydrogenase it's one of, there are several different markers for breast cancer stem cells. It's one of the markers.
And so the colon cancer, were those also sorted by aldofluor?
They were sorted by LGR5 positivity.
Okay. Did you try any other cancers that were sorted by aldofluor or ALDH
Not
positivity?
Not yet. This, we actually have a little grant to continue to work on trying to get different types of cancer stem cells while we are investigating the breast cancer. It, just like what Ken's lab did, where they looked at concordance in order to test putative pathways and identify genes, that's what we're doing now between breast cancer stem cells and the zebrafish. And so far, we have a couple of hits, and they're both very interesting. So. Thank you.